Hello, 109 lab students. This is Dr. Jenkins, and we are back. So we had 2 weeks to go over the muscles, and today we are doing, or this week, we are doing the nervous system. So here we go. You are going to begin your lab by reviewing the two main parts of the nervous system. I know that you talked about this in a lecture, but we can always review. The more reviewing, the better. So of course we have the central nervous system, which is made up of the brain and spinal cord. And this is the part of the nervous system that does, processing and integration. And then we have the peripheral nervous system, which consists of all of our peripheral nerves. And the function of the peripheral nerves is to transmit nerve impulses. So in those peripheral nerves, there is not any processing going on. The peripheral nerves only send and receive nerve impulses, whereas the central nervous system, the brain and spinal cord, is where we do the processing. Excellent. Then you are going to review all the anatomy, all the beautiful anatomy. You're going to start with the outer layer, or the outer covering of the brain and spinal cord, called the meninges. And I want to point out that this covering is continuous and it goes over both the brain and spinal cord. If I go to the next picture, you can see in this picture. So we have a covering that goes around the brain and the spinal cord, and then it comes back up and it continuously goes around. So it covers both. The meninges collectively has 3 layers, all right? The outer layer is called the dura mater. It does technically have 2 layers, but we're just gonna consider the dura mater, what I'm coloring here in red. You know, of course we have the skin and then the skull, and this is the layer right under the skin and the skull. Then we have an arachnoid space or an arachnoid matter. So it's this space here. It gets its name because it has these little protein filaments that kind of look like a spider web, hence arachnoid. And what's cool about this is that in the arachnoid, that's where the blood vessels come through. Lastly, let's pick a blue color. We have the innermost layer, which is called the pia mater. The pia mater is thin and delicate, and it clings right to the surface of the brain. If I was going to give you an idea of what these layers really feel like, the dura mater, the outer matter, is tough. It's almost like a leather, kind of. Not quite that thick, but it's really tough and thicker dura mater. The pia mater is almost like a saran wrap covering. It's thin and delicate, but the pia mater conforms to every nook and cranny of the brain. And you can see I've written out these right there for you. Next, you're going to learn the actual parts of the brain. So I find it easiest to break the brain down into 4 regions. So what I'm going to color in red here, the biggest part of the brain, the wormy part, so to speak, is called the cerebrum. The little cauliflower-looking thing on the posterior aspect is called the cerebellum. The inner part, so if I was to kind of put a little rectangle in here, this inner part is called the diencephalon. And lastly, let's go with a gray. No, because the actual picture is gray, Dr. Jenkins. Way to go. How about purple? The actual stem of the brain is aptly called the brainstem. So I would start there by learning the 4 regions or 4 sections. And then you're going to go over the anatomy based on these 4 sections: cerebrum, cerebellum, brainstem, diencephalon. Before we do that, there are some outer structures for you to identify. We have some cranial nerves. So the olfactory nerve, I think it's a better picture. Let me see here. They have the olfactory bulb. I'm just going to draw it in, folks. The olfactory nerve is a nerve that runs straight up and down. So we're looking at the underside of the brain. So let's call these the olfactory nerves. And at the tip, especially in the sheep brain, which we're going to watch a video of a dissection of the sheep brain, at the tip is a thicker little region called a bulb. Sheep are going to have a bigger olfactory bulb than humans, simply because sheep rely more on their sense of smell than we do. Then we're going to have the optic nerves. So we can see here's an optic nerve and here's an optic nerve. These are, of course, going to go to the eyeball. And then where the optic nerves come together is called the optic chiasm. So if we were to look at this up close, you have the optic nerves coming in and out. So this would be, in the person, this would be their right side, their left side, and these nerves would go to the left eye and to the right eye. The region where they come together in the brain, so the region where they come together is called the optic chiasm. Excellent. Excellent, excellent. Now let's continue. So if we look at the cerebellum, the cerebrum. So you remember the cerebrum is this wormy part, right? Well, this is a top view. It has two hemispheres, and those two hemispheres are connected by a little space, and that space is called the longitudinal fissure. If we look at the surface or the outer edge of the cerebrum, the outer edge is called the cerebral cortex, and I'm going to go back. So just the outer region, so just what I'm drawing in red here is the cerebral cortex, because indeed, cortex means outer. So in many organs of our body, we have the renal cortex. Well, that's the outer region of the kidneys. Here, the cerebral cortex is the outer region of the cerebrum. Along that outer region of the cerebrum, that wormy surface kind of goes down, then back up, down and back up. We can see it on this picture. Maybe I'll use a different color. I want you to be able to see it, folks. Yes, purple. So we have these crevices, right? And if I go back to this picture, let me use a color you can better see. Along the cerebral cortex, it kind of goes up and down. And the reason it does that is because it gives us more surface area. Well, if we're more specific here, every depression is called a sulcus or sulci. And the way that I think about this is if I'm depressed, I might sulk. So the depressions are called the sulci. And then the parts that are the ridges are called it's called a gyrus or a gyri. So it would be the elevation. All right? It is shown on one of these pictures when you look through the labs. But the purpose of that is for more surface area. The last thing here for the cerebrum would be the corpus callosum. The corpus callosum, I'm surprised it's not labeled there. Let's see, was it on this one? Yeah, it is labeled on this one. The corpus callosum, let me just clear this out for us. I love this pen. You know, it's one of the iPad pens, and I was hesitant that it was going to be able to draw well, but actually I think it's doing okay. So the optic chiasm, let's go to the pen. As long as I use it correctly, it'll work all right. The optic chiasm is this structure right there. It's kind of an arc shaped, right? It's shaped like this. The corpus callosum. The corpus callosum is functionally, I'm not going to ask you the function, but the corpus callosum is what allows information from one cerebral hemisphere to be communicated to the other. But anatomically speaking, it's just this center arch. All right, let's move to the cerebellum. The cerebellum is this whole back cauliflower looking thing. And then inside the cerebellum is a pattern of white matter called the arbor vitae. So what I'm drawing in, this pattern, is called the arbor vitae. It gets that name because arbor means tree in Latin, and vitae means life. So it gets its name because it resembles a tree of life. So the whole structure is called the cerebellum, but just the inner pattern of white matter is called the arbor vitae. Looking at the brainstem, we have 3 regions, 1, 2, 3, and these are listed from top to bottom. And these are simply that, regions. So we can look at the side picture. So we have the midbrain, which is the upper region, midbrain, pons, next region down, and medulla is lowest region. They're continuous, but the midbrain would be the highest, pons, and then medulla. You can kind of see it in this bottom picture, but not so well. So I would ask you on a side view like this. So if I add this, this would be midbrain. Midbrain is the highest or most superior, pons, middle region. And the pons you can tell also because it's the only one that kind of comes out a little bit. And then the medulla is the lowest one. And the medulla will blend right into the spinal cord. It's not like there's a line where we know exactly, but the medulla will blend into and become the spinal cord. Lastly, we have the diencephalon, which is that inner region. And let's talk about those. I'll use this picture. You know, it's easy to think that the brain is going to be really hard. And of course the brain is complex. But I'm really asking you to know only some of the basic structures. So before you even shove your homework aside and think, oh, I can't get this, you can. You just have to be patient. And I'm asking you really main structures here. So nothing too specific that you can't handle. All right, let's talk about the diencephalon. So the diencephalon, as we said, I'll use red, is this inner region, right? So if we look more specifically, we're going to see a central part called the thalamus. It is a region. When I teach this in person and I have the actual brain model, we would look at this whole region as the thalamus. When I have it in person, I almost say it looks like a boob because it's an elevated oval and it has this elevated ridge on it, kind of like a nipple. People laugh, but if you can think of any way to remember these, do it. Don't be shy. So that region in red is going to be the thalamus. Then we have a region in front of it and below it called the hypothalamus. So the thalamus is more central, and then in front of it and a little below it is the hypothalamus. Lastly, we have 2 glands. Hanging down from the hypothalamus is a gland called the pituitary gland. Really important gland. It is the gland actually in our body that releases the most hormones. It's crazy, that little ball that hangs down from your hypothalamus. Pituitary gland. And then behind the thalamus, we have another gland called the pineal gland. So just be careful because they both begin with a P, but the pineal gland is behind the thalamus. It actually secretes a hormone called melatonin. which you may be familiar with because it deals with our circadian rhythm and our sleep-wake cycle. Okay, everyone, you can do this with the anatomy. I'd like you to watch the sheep brain dissection. It's pretty cool. In the dissection, it kind of looks like a pile of mush, really. But if you work with the tips that I've given you and you focus on the regions, I know you can do it. The second part of this lab is dealing with the reflex arc. And this is really important. We rely on our reflexes because they are a major source of protection. Make sure that you know that function, protection. An easy example would be our pupil reflex. So as we're going to see, and you may already know this, if we shine a light near someone's eye, or if you go outside on a sunny day, what's going to happen to your pupils? They're going to constrict. And it's a way of protecting your eyeball from the light. Protection. A reflex itself is just defined as a quick response to a stimulus. What makes it a reflex is that it is super, super quick. So quick, in fact, that you don't have to think about it first. You know about a reflex. You don't have to think about your pupils constricting. They do it for you. You don't have to think about your leg kicking out when we strike the patellar tendon, right? So it's a quick response to a stimulus, so quick that it's involuntary, and it is really good at protecting us because it's so quick. Make sure you review the 5 components of the reflex arc. Here they are listed. And then I also give you a picture. This is actually from your lab manual. The first step is some kind of receptor. So imagine in this example of a reflex, imagine a hand here. Really bad drawing coming, watch out. 1, 2, 3, 4, 5. There's a hand. And you touch something sharp. Well, if you touch something sharp, what is your reflexive action going to be? Your reflex is going to be to pull your hand back. That's a reflex. You touch something sharp, your body's going to pull your hand back as a way of protection. And it happens without you realizing it. It's not like you keep your hand on that sharp object and wait until your brain processes. Oh, wait. That feels sharp. Maybe I should pull it back. No. You don't have to go through that process. It's a reflex. It happens for you. So the receptor would be whatever, in this case, whatever sensory receptors you have in your fingers. So the receptor is what senses the stimulus. What senses the stimulus? It senses the bright light. It senses the sharp object. A receptor is, it's usually a nerve ending that senses the stimulus. Then your nervous system will send a sensory neuron. Oh, that's the wrong way, Dr. J. Oh, that rhymed. Wrong way, Dr. J. We're going to send a sensory message to the spinal cord. You may remember from lecture, sensory nerve impulses travel from the periphery to the brain or spinal cord. Step 3, so that's step 2. Step 3 is going to be processing. Sometimes processing. How about I cannot spell and write at the same time today? Processing. The processing will sometimes happen in the spinal cord, other times it will happen in the lower parts of the brain. But for our intents and purposes, we're going to say that the processing happens in the spinal cord. This is why it's so fast. We don't have to wait for that nerve impulse to travel from the spinal cord up to the brain and then back down. We can process right in the spinal cord, and that's why it's so fast. Then we're going to have a motor impulse leaving the brain out to the body. And you can remember a motor neuron or a motor impulse sends information from the brain back out to the body. And our last step is going to be the effector. This is typically a muscle, and it changes based on what reflex you're talking about. But here, it would be the muscle that pulls your hand away. Muscle that pulls your hand away. In the pupillary reflex, it's the muscles that change the size of the pupil. It's the muscle that causes the response. Cool. Now, if we were in lab, we'd be doing this in person. We can't, so I've got some videos and you can go through it on your own. We have the patellar tendon reflex, so you're going to go through what that is and watch the video. We have the Babinski or plantar reflex. This involves using the sharp end of the reflex hammer to draw a C pattern on the bottom of the foot. And you can read about what the expected response is in an adult and in a baby. And then lastly, we have the pupil reflex. And there's a video at the end. When you are done with this video, you are going to complete the lab assignment and take the lab quiz. And then you're going to celebrate when you get a good grade, however you want to celebrate. All right, folks, see you next week.